Good morning everyone. Today in this class we are going to discuss about the linkage. Genetic linkage is the tendency of DNA sequences that are close together on a chromosome to be inherited together during the meiosis phase of sexual reproduction. When two or more genes reside on the same chromosome, they are said to be linked genes. Linked genes tends to stay together and inherit together during the formation of gametes. Linkage is the prominent exception of the Mendel's law of independent assortment. According to the Mendel's law of independent assortment, assortment of one gene occurs independently of the other non-allelic gene at the time of gametogenesis. Because of such an independent assortment of non-allelic genes, new combinations of characters are produced in the offsprings. In case of linkage, there won't be any assortment of these two genes, hence we cannot see the new combinations in the offspring generation. In offspring generation, only parental combinations can be seen. The first experiment to demonstrate linkage was carried out in 1905 by William Bateson and R.C. Punnett. Coupling and repulsion theory. Coupling is the condition where the genes that are coming from the identical parents enter the same gametes are inherited together. In repulsion, the genes are coming from the different parents and therefore separated in different gametes which are separately inherited. Chromosome theory of linkage. Boveri and Sutton proposed chromosomal theory of linkage. According to the chromosomal theory of linkage, genes that show linkage are situated on the same chromosomes. Genes are arranged in a linear fashion in a chromosomes and each gene occupies a definitive locus in a chromosomes. The linked genes inherit together during the process of gametogenesis. The distance between the linked genes is inversely proportional to the strength of linkage. The closer the distance, the stronger is the linkage strength, and the distance between the genes increases, thus linkage strength will decrease. Linkage group. All the genes present on a particular chromosomes form a linkage group. The number of linkage groups of a species corresponds to the total number of different chromosomes of that species. It is not simply the number of upright chromosomes in an organism, but it is a different chromosomes present in that species. For example, in humans, there are 23 pair of chromosomes, 22 autosomes and 2 sex chromosomes. In males, there are 22 autosomes, 1 X and 1 Y chromosomes. So, the total number of linkage groups are 24. Since X and Y chromosomes are not homologous chromosomes, they form a two different linkage groups. Hence, the total number of linkage groups are 24. In case of females, they have 22 autosomes and 2 X chromosomes, and these two X chromosomes are the homologous groups and form the same linkage group. Hence, the total number of linkage groups present in human female are 23. Types of linkage. There are two types of linkage, that is complete linkage and incomplete linkage. Complete linkage is when the genes are closely associated to each other. They tend to remain together and have no chance of separation from each other during the inheritance. This phenomenon is called as complete linkage. In complete linkage, the parental combinations of characters appear together for two or more generations in a continuous and regular fashion. Example of the continuous linkage is grey body and long wing in the male Drosophila. Incomplete linkage. In incomplete linkage, the linked genes which are located in the farther distance in a chromosomes, do not always remains together and separate during the gametogenesis. This type of linkage is known as incomplete linkage. Incomplete linkage is due to the crossing over between the homologous non-sister chromatids during the prophase 1 of meiosis. Example: white eye and miniature wing in the female Drosophila. Incomplete linkage depends on the distance between those two genes. If the distance is more, then the chances of getting recombination is also more. Thank you.